morning, I am Madhurima Datta, Zoology and Biology educational content creator. Now's content for B.Sc and M.Sc Zoology, Microbiology Honours, Physiology Honours, Biochemistry Honours and also Microbiology Honours. And also exam for CVT PG exam, GATE exam, DBT JRF exam, IIT JAM exam and CSI NET Life Sciences exam. Paper Molecular Biology, Unit 1, Introduction of Molecular Biology, and today's topic is nucleotide, nucleoside and introduction of some molecular biology. Let's go. The nature of genetic material. The studies that eventually revealed the chemistry of genes began in Tübingen, Germany in 1869. There, Frederick Meister isolated nuclei from pus cells. This is the white blood cells in waste surgical bandages. He found that this nuclei contained a novel phosphorus-bearing substances that he named nuclein. Next, structural component of the genetic material. A complete nucleotide consists of 3 covalently bonded units, a nitrogenous base, pentose sugar, and 1 or more phosphate groups. And a nucleoside consists only of the nitrogenous base and pentose sugar. Next, nitrogenous base. Nitrogenous bases are planar, aromatic, heterocyclic compounds divided into major classes. First is the purines, which is bicyclic, consists of fused pyrimidine and imidazole ring, that is 9-membered ring system. Example of the purines are adenine, that is 6-aminopurine, and guanine, their chemical name is 2-amino-6-oxypurine. Next, pyrimidines. This is a monocyclic ring, consists of a single 6-membered heterocyclic ring. Pyrimidines components are cytosine, uracil and thymine. The chemical name of cytosine is 2-oxy-4-aminopyrimidine. Chemical name of uridine is 2,4-dioxypyrimidine. And chemical name of thymine is 2,4-dioxy-5-methylpyrimidine. It is also called 5-methyl-uracil, which is present in DNA. Next, tautomerism. Nitrogenous base exist in equilibrium tautomeric forms. First is amino versus imino. Adenine and cytosine predominantly exist in amino form at physiological. Their pH is 7.4. And next, keto versus enol group. Guanine, thymine and uracil predominantly exist in keto, that is lactam form, rather than the enol lactim form of physiological pH. This is critical for standard Watson-Crick hydrogen bonding. Next is short note of pentose sugar. The sugar unit is a 5-carbon aldopentose in its furanose ring form. First is beta-D ribofuranose. Present in ribonucleotides, that is RNA, possesses hydroxyl OH groups at both C2' and C3' end. Next, beta-D2' deoxyribofuranose, which is present in deoxyribonucleotides, lacks an oxygen atom at C2' position, having a hydrogen atom instead. Next, sugar puckering or sugar conformation. The furanose ring is nonpolar to minimize steric strengths among substituents. 1 or 2 atoms shift out of the 4-atom plane. Next, C2' endo. C2' lies on the same side of the ring plane as C5', characteristics of beta DNA. Next, C3' endo. C3' lies on the same side of the ring plane as C5', which is characteristics of aDNA and RNA. Next, the chemical nature of polynucleotides. By the mid-1940s, biochemists knew the fundamental chemical structures of DNA and RNA. When they broke DNA into its component parts, they found this constitute to be a nitrogenous base, phosphoric acid and the sugar deoxyribose. Next point, RNA yielded base and phosphoric acid plus a different sugar ribose. The 4 bases found in DNA are adenine, cytosine, guanine and thymine. Next, RNA contains the same bases except the uracil replaces the thymine. Adenine bonds to thymine and cytosine bonds to guanine. Adenine and guanine are related to the present molecule purine, which we refer to this computes as purines. Where ribose contains a hydroxyl group in the 2 position, that is deoxyribose lacks the oxygen and simply has a hydrogen, represented by the vertical line, hence the name deoxyribose. The bases and sugars in RNA and DNA are joined together into units called the nucleosides. Because thymine is not usually found in RNA and deoxy, which is designation for its nucleoside is frequently assumed, and the deoxynucleoside is simply called the thymidine. Next, the base is linked to the 1' position of the sugar and the 2' position is deoxy in deoxynucleosides, and the sugars are linked together in DNA and RNA through their 3' and 5' positions. The base is linked to the 1' position of the sugar. The 2' position is deoxy in deoxynucleosides, and the sugars are linked together in DNA and RNA through their 3' and 5' positions. Next, the subunits of DNA and RNA are nucleotides, which are nucleosides with a phosphate group attached through a phosphoester bond. Next, it is an important question for major honors in molecular biology or zoology or microbiology or physiology or biochemistry. That is, why does we call the ester of the DNA a phosphoester? Because an ester is an organic compound formed from an alcohol which bearing a hydroxyl group and an acid. In the case of a nucleotide, the alcohol group is the 5' hydroxyl group of the sugar and the acid is phosphoric acid. So, when synthesis of DNA takes place, 2 phosphate groups are removed from DATP, leaving deoxyadenosine 5' monophosphate or DAMP. The other 3 nucleotides in DNA, that is DCMP, DGMP and DTMP, have analogous structure and names. And the structure of the bond that join nucleotide together in the DNA and RNA, these are called phosphodiester bonds because they involve phosphoric acid linked to 2 sugars. One through a sugar that is 5' position and the other is through a sugar that is 3' group. This more closely resembles their geometry in DNA or RNA. Note also that this trinucleotides or string 3 nucleotides has polarity. The top of the molecule bears a free 5' phosphate group, so it is called the 5' end. The bottom with the free, that is the 3 hydroxyl group, it is called the 3' end. Now the topic has ended. For all video of B.Sc and M.Sc in life science in major and minor, please subscribe my YouTube channel Biogenesis by Madhurima and follow my Facebook account Madhurima Dotto.